A decades-long boom in brain science in the United States may be heading for a bust. Ongoing disruptions in federal funding are causing many young neuroscientists to reconsider their careers. NPR's John Hamilton reports that losing these scientists could slow research on everything from autism to Alzheimer's.
About 20,000 brain scientists are expected to gather in San Diego this weekend for the annual meeting of the Society for Neuroscience. But Clara Zundel, a postdoctoral researcher at Wayne State University, won't be there.
Because of the funding changes, I had to consider travel costs this year.
Zundel studies how pollution affects the developing brain. Her work is funded by the National Institutes of Health. But since President Trump took office, the NIH has been buffeted by cuts, grant terminations, and abrupt policy changes. So Zundel is unsure whether support for her research will continue or if she'll be able to find a steady job.
Many universities are still on partial or even full hiring freezes. And so it's just made it really, really scary to think about how I'm going to take that next step.
Even so, Zundel isn't ready to abandon her career plans just yet.
Talk to me in another 3 months, and I might change my mind. But I absolutely love what I do. I want to continue doing what I do, and I want to continue to do that here in the United States.
Other young researchers are less certain, says John Morrison, a professor at the University of California, Davis, and president of the Society for Neuroscience.
You hear things like, I've prepared my whole life for this. Is it gone now? Is it no longer possible to be the scientist that I always wanted to be? Many will just choose something else.
Or take their research to another country. Morrison says what's at stake is the next generation of scientists who will study the circuitry underlying disorders like Alzheimer's, Parkinson's, and schizophrenia.
The U.S. has been a world leader in research for decades, and that leadership position is now at risk.
The NIH often awards 5-year grants, and scientists tend to structure their research around that timeline. But Morrison says many awarded grants have been paused or summarily terminated by the Trump administration.
If you disrupt the grant in the middle of it, you're going to disrupt that whole progression. And you'll get to a point where the work that you've already done is worthless.
Federal health officials say the cuts reflect an effort to reduce waste, end support of woke science, and align research with the administration's priorities. Morrison says cuts can produce short-term savings, but in the long run, he says, the human and financial costs will be huge.
We often quote Mary Lasker, who was a renowned advocate for biomedical research and launched the Lasker Prize. And she said, if you think research is expensive, try disease.
Brain science has long enjoyed bipartisan support in Congress. Diane Lipscomb, a professor at Brown University, is chair of government and public affairs at the Society for Neuroscience. She says one reason lawmakers support basic research is that ever since World War II, it has been a huge boost to the U.S. economy.
I don't think we've ever talked to anyone in the Congress who disagreed with that.
But cuts and disruptions have come from the executive branch, not Congress. So neuroscientists are taking their case directly to the public. The Society's website, for example, now includes videos of scientists explaining what they do and why it matters. Lipscomb thinks that message will be heard, and when she talks to young scientists asking for career advice, she tries to offer an optimistic message.
You just have to stay with what you know is what you love, because things will get better.
At least that's the hope. John Hamilton, NPR News.
Hello, I'm John Morrison, and welcome to the presentation of the 21st Annual Gruber Neuroscience Prize, on which the Society for Neuroscience is pleased to collaborate. The Gruber Foundation presents 3 annual $500,000 prizes in the fields of cosmology, genetics, and neuroscience. Sarah Rea, the executive director of the foundation, will say more. Sarah?
Thank you, Dr. Morrison. We are delighted to present the Neuroscience Prize at SFN's 54th annual meeting and to continue to work with the Society despite the fact that Marty Sagesi is leaving. The Gruber International Prize Program, established in 2000, recognizes achievements and discoveries that produce fundamental shifts in knowledge and culture. While we are here to honor Edward F. Chang, Let me mention that he joins Cosmology Prize recipients Ryan Cook and Max Patini and Genetics Prize recipients Rotem Sorik on our 2025 roster. Please note that nominations to the 2026 Gruber Prizes are open until December 15, 2025, and that we encourage nominations that reflect the breadth of the fields and the diversity of those working within them. And before we return to neuroscience, I simply must acknowledge our co-founders, Peter and Patricia Gruber, whose combined vision and leadership established the International Prize Program, and whose care in doing so gave it the legs to stand on its own. We are grateful to SFN for its support of the Gruber Prize, but also for administering the International Research Award in Neuroscience that we fund. Now let me ask John Morrison to return to present the year's awardees with their plaques.
Thank you, Sarah. It is now my honor to present the Peter and Patricia Gruber International Research Award in Neuroscience. This award recognizes up to 3 promising young neuroscientists for outstanding research and educational pursuits in an international setting. This year's recipients are Dr. Lingxiao Xiao, Dr. Maitreyi Wairagkar, and Dr. Ipshita Zucchi. Dr. Lingxiao is a postdoctoral fellowship at Cornell University. She attained her PhD at Zhejiang University in China, where she studied the pathophysiological basis of anxiety disorders. At Cornell, her research focuses on how psychoactive compounds such as ketamine and psilocybin can remodel neurocircuitry. Her contributions, detailed in 2 lead author publications and 6 co-authored papers, have advanced the understanding of how psychedelics reshape brain circuits and may inspire new therapeutic approaches. Please join me in welcoming Dr. Xiao. Our next recipient is Dr. Maitreyi Wairagkar. Dr. Wairagkar is a postdoctoral fellow at the University of California, Davis. Go UC Davis. Where she studies medical neuroprostheses to restore speech in individuals with neurological injuries or neurodegenerative diseases. Born in India, she completed her undergraduate and graduate studies in the UK. In her first postdoctoral position at University College London, she developed affective social robots and conversational AI to help people with dementia. And she is now developing intracortical brain-computer interfaces for restoring speech. Please join me in welcoming Dr. Wairagkar. Our third recipient is Dr. Ipshita Zucchi. Dr. Zucchi is a postdoctoral fellow at NYU Langone Medical Center, where she studies how hippocampal circuits interact with the cortex to transform sensory inputs into internal signals that support planning, decision-making, and memory during goal-directed navigation. In her recent work, she designed an innovative auditory-guided navigation task in mice and found that neuronal activity was driven more strongly by preparation for action than by external sensory inputs. Please join me in welcoming Dr. Zucchi. Now you want... Okay. And Sarah, I believe, there you are.
Congratulations to our 3 2025 awardees, and thanks again, Dr. Morrison. We hope to see you 3 awardees back here someday, perhaps receiving the Gruber Prize in a few years. The 2025 Neuroscience Prize recipient was chosen by our Distinguished Selection Advisory Board. These volunteer advisors bring their priceless expertise and their commitment to the selection process, along with an irrepressible enthusiasm for science, and they are nominated to serve by SFN. We couldn't do this without them. Let me now ask our board chair, Joshua Sains, to read the official citation to the prize drafted by this board and add any further remarks. Professor Sains.
Thank you. It's a pleasure to read to you the official prize citation. It reads, the Gruber Foundation proudly presents the 2025 Gruber Neuroscience Prize to Edward F. Chang for his fundamental research in functional brain mapping, observing electrical patterns in the brain related to speech. Dr. Chang's research has helped to map specific brain regions involved in understanding the assembly and processing of speech, the processing of pitch, rhythm, and intonation, and more. This work has involved analysis of the collaboration between specific motor regions in the vocal tract and how these systems work together in speech processes. Through his research findings, Dr. Chang has contributed to a clinical trial aimed at addressing speech impairments caused by paralysis in human populations using ingenious brain-machine interfaces with cortical input. So let me just finish with a few words about what it means to me. Like most of you, I've spent my life working on model systems. These are accessible preparations that we think will eventually help us to understand what we want to understand most, which for most of us, including me, is the human brain. That's how I got into it. I bet many of you got into it for that reason as well, how the brain works and how sometimes the human brain doesn't work quite properly. And this has been a spectacularly successful approach, as probably everybody here knows. But Eddie has taken on the challenge of studying the human brain itself. Moreover, he's focused on one of rather few areas that can only be studied in the human brain, language. Really an interesting and brilliant choice. And so he is now one of the pioneers at the forefront of what I believe will be one of the most exciting areas of neuroscience over the next few decades. And I look forward to hearing him tell us about what he's been doing. Eddie.
There will be a moderated discussion with our prize recipient after his lecture. The moderators are Eve Martyr and John Monsell, who are advisors to the prize.
I'm extraordinarily honored to be a recipient of the 2025 Gruber Foundation Prize in Neuroscience. It's particularly prestigious in terms of its recognition in a global field that I've had the pleasure to train in for my entire career. And I thank the committee and many of my mentors here and lab that have participated in this research. So the focus of this lecture is really on the evolving neurobiology of our ability to speak. The title is The Neural Code of Speech. I wanted to take this lecture to really explain the opportunity that I've had as a clinician scientist, in particular as a neurosurgeon that takes care of patients with brain tumors, epilepsy, movement disorders. It's through this privilege as a physician that I've been able to interact directly with the human brain, both deep and in the cortical layers. And this has been an extraordinary opportunity to directly study the substrates of human cognition. And the system that I've been most interested in is trying to understand the biology of words. Our research involves the implantation of electrode arrays, sometimes that we record temporarily in the operating room. Other cases will implant for 2 to 3 weeks for the localization of seizures. And it's in that setting that we've been able to access the human brain with unprecedented temporal and spatial resolution in a way that is otherwise impossible to do. So this has been an important opportunity in order to directly study from the human brain itself. The technology by itself, however, is not sufficient. The most important critical ingredient for doing human neuroscience really is actually the partnership with the patients that are involved in this work. So everything that I'm about to show you has really resulted from an incredible partnership between scientists, physicians, and the patients that have dedicated their time to participate in these trials. This is what a typical recording session looks like in our lab. 2 of our former graduate students, Leah and David, are recording direct brain signals from a 256-channel sensor array from one of our epilepsy patients' brain. And you can see the patient's mom looking on. This is really a full partnership between patients, their families, and our research team. And in this setting, in addition to streaming hundreds of channels directly from the cortical surface, we're able to engage participants in tasks, speaking, listening, memory, and more. The topic of this lecture pertains to a specific, unique, and defining human behavior, which is our ability to speak. The segments that I'm going to describe have to do with speech perception, our ability to understand sounds and how that's transformed into words, then how we produce words, how those discoveries have led to the development of the first speech neuroprosthetic devices, and then also ongoing work to discover the cellular basis of language. I'm starting with the topic of speech perception, which is how sounds are transformed into words in the cerebral cortex. This video basically shows the electrocorticography sensor arrays that are located over the superior temporal gyrus in a patient that's undergoing monitoring for seizures. These methods still are the most resolved methods that we have for measuring both the millisecond and millimeter resolution from the human brain. And what you're going to see in this video is the time-lapse activity within this particular part of the brain when someone hears a sentence.
And what eyes they were.
You can see that five words in a very fast sentence, less than 1 second, happens in less than a blink of an eye, which is why we need this millisecond resolution. When we can slow down this activity down to the order of milliseconds, then we can resolve what is happening at each one of these individual positions.
And what eyes they were.
And for the rest of this lecture, what I'm going to be describing is how we've been able to interpret the electrical activity of the human auditory cortex, also known as the Wernicke's area, and how it has given rise to our ability of how consonants, vowels, and all of the phonetic inventory of language have been encoded there. Our first huge window into this was a paper that we published in 2014 with my former postdoctoral fellow, Nima Mascarani. What you're looking at on the left is the electrode selectivity to all of the different consonants and vowels in English. And what you can see is this one particular electrode on the superior temporal gyrus is tuned to a series of consonants like da, ba, ga, pa, ta, ka. In linguistics, we call these stop consonants or plosives. If you look at the electrode next to it, it's tuned to a separate class of phonemes that we call fricatives, like the sh and the z sound. And another electrode tuned to low back vowels like ah. Here's one tuned to high fronted ones like e, some nasal sounds like na and ma. This was the first time that we could see that not only this part of the brain, the superior temporal gyrus, was activated by speech, but we could see what was happening locally in neural populations at a very fine millimeter resolution. And it wasn't just at a couple of electrodes. We could look at this over hundreds of electrodes and now tens of thousands of electrodes collected over the last 15 years and study how the electrical activity encoded at these particular sites give rise to all of the different sounds that we have in human speech. We refer to these as the building blocks of speech because by themselves they have no meaning. But in combination, they give rise to all possible meaning when we communicate. And one of the discoveries from this set of studies was that if you cluster the data and you look at its organization, you will see that it actually is tuned hierarchically to different categories linguistically that we know of as consonants and vowels and the different subclasses of those. And these are actually linked to a featural description of linguistic phonetic encoding in the superior temporal gyrus. There's another really important dimension of speech, which is the intonation of the voice. It's not just about the consonants and vowels, but actually the way that we change the pitch of our voice in order to convey meaning. And in English, I'll give you some examples of how changing the pitch on one particular word can change its meaning. You're looking at the same sentence, but in each one of these sentences, the pitch contour is placed on a specific word. In the first case, it's a neutral, where it's not particularly strong on any particular word.
Movies demand minimal energy.
In the second example, the fourth word is emphasized, and it changes it from a statement to a question. This is how we use the pitch of our voice.
Movies demand minimal energy.
In another example, if you stress the first word, it emphasizes movies and not something else.
Movies demand minimal energy.
In a fourth example where the stress is placed on the third word.
Movies demand minimal energy.
So we use pitch information and prosody intonation in order to convey nuanced meaning in English. And one of the discoveries from this study that was carried out by one of my graduate students, Claire Tang, was that if you look at the electrical activity at one particular electrode, as someone is speaking multiple sentences with stress at different words, you can see that the neural activity at that one particular electrode is following and tracing the pitch of the voice. It's not tuned to any of the particular words or the consonants and vowels within them, but actually is selective and independently selective to the pitch and voice of that speaker. And we looked at this for both female speakers, which tend to have the dynamic range of pitch of voice around 200 hertz, and also male speakers, which have the dynamic range of pitch of voice around 100 hertz. And despite the fact that these two speakers have completely different dynamic range of pitch in the voice, the same electrode is encoding activity for these two particular speakers. And one of the discoveries that Claire made in this study was that it's not about the absolute pitch of the voice, but it's actually the change in the pitch and the trajectory of the change of the pitch, which is what is encoding at these particular sites, not the absolute pitch itself. In a different study carried out by Liberty Hamilton in my lab, we also had the ability to introduce electrical recordings not just from the superior temporal gyrus, which lies on the lateral aspect of the temporal lobe, but also covering the deeper medial temporal lobe, which is buried within the sylvian fissure, which requires a surgical procedure in order to open up the sylvian fissure to have access to the sensors. And in a select set of cases, we had that opportunity to explore the entire human auditory cortex, from the primary auditory cortex in the medial Heschel's gyrus to the lateral associative auditory cortex in the superior temporal gyrus. And this is a 3D reconstruction of what those recordings look like, sampling from several individuals across the entire human auditory cortex. One of the questions that we wanted to understand was, what is the nature of information flow as information is transformed from the spectrotemporal pattern of sounds into the linguistic representation of phonetics and words. And what we found in this study was actually quite interesting and different than what we predicted. What we found basically was that there is parallel, not serial functional organization of the auditory cortex, which is very different than how we think about the visual system. In this particular paper, we described through various lines of analysis how activations in the human primary auditory cortex are different but parallel from those that we see in the STG. In the primary auditory cortex, the activation to speech is basically equivalent to that responding to pure tones. Its organization is tonotopic. In the lateral superior temporal gyrus, the associative auditory cortex, the responses to speech are far greater, 3 to 5 times greater than they are to pure tones. And its organization cannot be understood by tuning to pure tones. However, what really surprised us was when we started to look at stimulation and the effect of resections. We found a double dissociation. When you stimulate the medial Heschel's gyrus, the primary auditory cortex, it evokes sound hallucination. And interestingly, there is no effect on speech perception when the primary auditory cortex is ablated or removed. There is normal speech perception when the primary auditory cortex is removed. And that's in stark comparison to what we find in the lateral temporal cortex, in the planum temporale and the lateral STG. When you stimulate there, it does not evoke any sensation. No one reports hearing anything. However, when you stimulate while they're listening to words, it actually impairs their ability to perceive it as speech and words. In fact, if you remove that area, or if you have an injury to that particular area, it will cause a syndrome that we called word deafness. And so in this particular paper, one of the interesting and important discoveries was really the observation of a parallel functional organization of the human auditory cortex. And we now know from labeling studies that the projections to these different parts of the auditory cortex are in fact independent. The projections to the medial auditory cortex are from the ventral part of the thalamus, the auditory thalamus, where the superior dorsal and medial parts of the thalamus project to the STG. So there may be these parallel pathways even at the level of the thalamus. Now, understanding the phonetic elements and intonation is just the building blocks of how we construct words and sentences. But the way that I'm speaking right now is continuous speech. And one of the things we've been really interested in is trying to understand the representation of words, essentially a sound image of an object that takes more information than just the segmental information of consonants and vowels, but one that helps us directly link between sound and meaning. This is what we call the auditory word form. It's the phonological referencing both the phonetic, prosodic, and also lexical representation of the sound, but not its meaning, just describing the sound image of the word. This is the key link between trying to understand how sounds can become objects that then later become linked to meaning. There are a couple of criteria or features that when you think about an auditory word form. One is that when you're listening to continuous speech, that continuous speech should be segmented into individual, distinct, recognizable words, not just a continuous stream of auditory activity. We should see evidence that the neural activity is time-aligned to the beginning and end of words, not other linguistic units. That there's information about the whole word above and beyond its sub-lexical features, and that it has certain kind of invariances for word duration, like we see in vision. And then also, last, that this would be something that would be highly influenced by our experience. So I'm going to walk you through how we've been able to see some of these criteria in order to observe a representation of the word form. Continuous speech lacks reliable cues for word boundaries. If you look at the text at the top, we don't really have those cues when you're listening to speech. We take that actually for granted as listeners. Unlike in writing text where we put spaces in order to really help the visual system distinguish between words, the auditory system does not have access to reliable cues, and certainly not pauses or spaces between every word. I'm going to play a sentence here, and I want you to appreciate how remarkable that is, that you're hearing words, not necessarily the elements that compose the words. This is the demarcations that occur between individual words, and these are notoriously lacking reliable acoustic or spectrotemporal boundaries.
For last year's legislative approval of 35 new judgeships.
So you hear words, not just the phonetic elements. And so listeners must rapidly associate this kind of acoustic input with stored knowledge. This is something that we learn. And this stored knowledge is about the phonological, the sound properties of words, and also the word knowledge that we have about them in a given language. In a paper that was just published last week in Neuron by Yizhen Zhang, postdoctoral fellow and a faculty in our group, and close collaborator Matt Leonard, described the first thing that I'd ever seen that was corresponding to word length units in human neural activity. This is something that was very elusive to us. We were looking for the word form area of the human brain. And what we found was that within the superior temporal gyrus, the same area that we see the coding of consonants and vowels, interestingly, if you look at the boundary of the neural activity across different word pairs that you see at the left, you see a pattern, which is the neural activity, in particular the high gamma activity, drops at the boundary point about 100 milliseconds between every word. This is evidence at the single trial and single word pair level of a chunking of the continuous speech signal at these different word boundaries. And this is what it looks like if you average the neural activity at different electrodes at the word boundary over hundreds and thousands of different electrodes. You can see that there is a clear, what we call an evoked response potential, where it increases at first, and then there's a very steep drop in the neural activity before it picks up again to the next word. And if you look at the distribution of the latencies across these what we call boundary electrode sensors, it's about 50 to about 150 milliseconds. So this happens at the word boundaries. The words that you're hearing from my mouth, in fact, in your own brain, if you understand what I'm saying, are actually being chunked in terms of the neural activity at word length units. And what's interestingly about it is the magnitude and the resetting or drop of that neural activity is directly correlated with the duration of the words, and not just the duration of the words, their lexical frequency and other lexical properties as well. So it has to do a lot with the word form property. And this is a simple schematic of what's happening. For the continuous speech stream, the neural activity is chugging along, but at each word boundary, you have a decrease in neural activity that kind of resets the activity and is scaled by the word duration and other lexical properties that occur throughout a word. Now, we did a very important control in order to see if this was driven by the acoustics or something that's driven by our knowledge of words. We think it's the latter, but we needed a more definitive control in order to prove this. And Matt created a stimulus that allowed us to do that. This continuous stream of words can be perceived as either boater or turbo, depending on what state of mind you are in. People tend to only hear one of these words, but not the other. And I'm going to play this to you, and I'd like you to participate in this particular experiment. If you hear the word boater, I want you to raise your left hand. If you hear the word turbo, I want you to raise your right hand. And what you will see is that at some point your perception may change. So I'll play that sound here.
Turbo, 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 turbo.
I think you get the point. And I saw someone dancing back there. So basically, what Matt discovered was that we can look at all those trials. We asked people what they heard on every one of those trials and when their perception switched from hearing boater or turbo or the other 6 word pairs that we looked at in this. And depending on what word you were perceiving, we could see a very clear switch in the timing and chunking of that neural activity based on how you were segmenting that continuous speech stimulus. It's the same stimulus, but just based on your brain state, how you're chunking that information. And this is an example that's completely controlled in terms of the acoustics, but has a very dramatic change on our perception. So there is this chunking of the auditory scene into different word length segments. A different experiment was carried out by one of my graduate students, Alina Bayer-Grossman, where she had particular interest in what is the consequence of language experience for people when they are exposed to it as a native. And over the last 15 years, we've had this really extraordinary opportunity to record from people's brains that really have different language histories. In San Francisco, we get exposed to folks who have, who are monolingual for Spanish, monolingual for English, and even monolingual for Mandarin. And part of these studies were actually done in collaboration with some of my colleagues in Shanghai. And so what we did in this particular study was we had people listen to their native language, and then we also had them listen to a foreign language, in some cases 2 foreign languages, whether it be Spanish, English, or Mandarin. And so what we wanted to look at is what's different when you have experience of a knowledge. And I'm going to play examples from this. Think about what you're hearing as you're hearing a language that you either understand or don't.
Siempre le fueron las cosas bien.
The wagons were burning fiercely.
大家试验了不同的方法。
So generally, when you understand it, you can detect the words like I just described. But when you don't understand a language, it sounds very fast and fleeting. We can still hear a lot of the phonetic elements, but we just can't make out what you would consider to be the word segments. So one of the things that Alina discovered is that if you look at the neural activity on both the left and the right side, where we do see activity to speech sounds, it basically looked identical between when people were listening to their native speech or whether it was foreign. Our prediction going into this study was actually that it was going to be a lot greater for your native language. That was our prediction. But in fact, what we found was a negative result. It was virtually identical in terms of the overall activation patterns. So whether you're listening to your native language or a foreign language, your superior temporal gyrus is activated generally just as much. And in fact, if you look at all of the different phonetic tuning, the things that I described early on, the different features of consonants and vowels, those were indistinguishable between the native and foreign speech, which means that those electrodes that are tuned to certain consonants in English are actually activated by you hearing those same sounds in a different language. The phonetic analyzer in the superior temporal gyrus is doing virtually or almost identically the same thing with a couple of small examples of the variance. So 95% of this is very similar between native and foreign speech. This was surprising to us, and therefore we were very careful to try to prove this as a negative, but it was very interesting that this was very similar. Now, at the same time that this study was being carried out, this other one that I had just described about word encoding in terms of the word boundaries was being done. And because of the cross-fertilization lab, we started to look at the word response that I described to you in this paper that was published just 2 weeks ago. And what we found was actually a quite striking difference at the level of words. That when you are listening to your native language, in fact, the superior temporal gyrus, even though it has the same overall magnitude, its chunking of the continuous speech is highly specific and very much depending on the word boundaries for your native language versus when you're listening to Ford. And this is expressed actually by the magnitude of these electrode responses that you see on the left. You can see it's much bigger than what we see in the foreign. And basically the thing that you see on the right is a schematic of what is going on. When you're listening to your native language, the ongoing speech stream is actually segmented along words. There's literally a drop in the high gamma neural activity at the different word boundaries. And when you're listening to foreign speech, it all becomes a continuous stream of information just being fluctually activated. So our human superior temporal gyruses are chunking this continuous speech stream into word-sized units. Alina also looked in the brains of people who are bilinguals, people who had equal proficiency in both Spanish and English. And what she discovered was that this word encoding was virtually identical, whether they were listening to their native, their L1 or L2 proficiency language. And she also found a gradient effect. If you look at the English proficiency, which is color-coded, we found that if all these different languages throughout the world, between Korean, Mandarin, Arabic, Spanish, Russian, etc., that there is a direct and linear relationship between your speech proficiency in English and how much it encodes this word boundary. So one of the key things that's happening when we learn our native language is that our brain becomes tuned to the statistics of the phonetic elements, the sequence statistics of it, way beyond what's actually available to us by the segmental phonetic information. And this has transformed our model of how we think about encoding of auditory information in the STG. This is a new model that we've proposed where this is multi-scale. It's not a serial feed-forward model, but really more of a recurrent multi-scale integrated kind of representation that has both the sublexical units of consonants and vowels, but also has information about the word form. And these kind of word form and lexical features are heavily influenced by the word boundary effect, sensitivity that we see to the word frequency, its duration, and we now know that this is highly experience dependent. So I'm going to switch gears to talking about a different system, which is how we produce words, the vocal motor control that allows us to shape the breath into words. One of the first studies that we did allowed us to resolve some of the basic aspects of the orofacial lateral motor cortex into the different representations of the larynx, the tongue, the jaw, a secondary laryngeal representation. These were things that we had assumed from the time of Wilder Penfield, but could never derive directly from speech because it really it needed this resolution of millimeter and millisecond temporal resolution. And in particular, what was a surprise to us, the discovery of this dorsal, more superior laryngeal representation, which is denoted by the X's. One of our graduate students, Ben Dichter, did his whole thesis actually trying to understand the representation of the dorsal laryngeal cortex. He had a task where people would say the same sentence, but actually stress different words across different trials. Like, I never said she stole my money versus I never said she stole my money. So again, this use of the intonation, the tension of the vocal folds is a critical function of the larynx. And what Ben found was that this dorsal part of the orofacial, the cortical bulbar part of the distribution of the homunculus, is exquisitely tuned to not the words themselves, but the intonation, the pitch that is occurring when you're stressing a particular word. And these electrodes were clustered in the dorsal part of the orofacial. We now call this the middle precentral gyrus because anatomically that's where it is, in the middle between the more dorsal and ventral areas. Some other interesting phenomena that he discovered is that when you're singing melodies, this part of the brain is encoding the pitch of do, re, mi. When you record those sounds and you play them back to people, these areas are encoding the auditory information, the pitch information of what you hear. And we now know that this is also an auditory cortex. It's part of the human motor system, but actually has auditory responses there. And also, to prove that this was causally involved in the control of the larynx, we stimulated electrically and could confirm that this actually evoked movement, EMG movement in the larynx. When someone's awake, it has a really interesting phenomenon. Every week on Tuesdays, I participate and lead awake brain surgeries at UCSF. And it's really one of the kind of procedures that got me interested in this as a career in the first place. There you're directly looking at the human brain and interfacing with it, trying to make sure that you can identify the areas that are critical for function, at the same time avoid those and protect those, and at the same time removing the spot of the tumor or the epilepsy in order to solve the pathology. And so what you're looking at is a bird's eye view of where I see over the surgical field, but then also the other side of the drape where our neuropsychologists, anesthesiologists work with our patients. And I'll show you this video from one of our early cases. These kind of experiences are an extraordinary privilege because we're right there at the interface between cognition and behavior. And what you're looking at is the exposed frontal lobe of an individual who's having a surgery for tumor. This was really one of the first videos that I had in my own career where we identified not only the areas that were involved with stopping speech, we call that speech arrest, and you'll see this in the video, but if we stimulate this dorsal laryngeal cortex or middle precentral gyrus, you'll see that it actually can evoke vocalization.
Count again?
We're going to count one more time.
Okay.
One more time, okay? So you can start at 1.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11,
right there.
12, a little tingling, okay. 13, 14.
Is it gone now?
A little bit on the tip of my tongue. 15, 16, 17, 18, 19. Now it's getting hard everywhere. 20, 21, 22, 23.
Yeah, he looks okay. Is it moving a lot or is it?
It just kind of tingled.
Still tingling?
Not now. It was a little hard to control.
All right.
24, 25, 20.
There we go.
26, 27, 28, 29, 30, 31, 32, 33,
okay, slurring that's an example of
34.
Vocalization. And it's that kind of mapping that we've actually had since the time of Penfield that's allowed us to get a gross idea of some of this organization, but it's actually the electrocorticography and the electrical recordings that really allow us to understand these functions. So when someone is awake and you stimulate that particular area, you will induce an involuntary vocalization. Now, there are other kind of movements that we care about, the ones that are above the larynx. So the larynx is what generates the voice, power, and energy. But then that energy is shaped by the resonances of the upper vocal tract. That's the lips, the tongue, jaw, etc. And this study that was carried out by Josh Chartier and Gopala, a postdoc in my lab, was essentially looking at all the different positions across the precentral gyrus and how those relate to movement trajectories, which we call speech gestures. They're not specifically just one articulator or another, but actually defined out and back coordinated movements. In particular, for this sound, duh, it's what we call coronal trajectory. And when you make that, the front of your tongue goes to the top of your upper teeth. The jaw goes up and down, the back of the tongue goes down. So it's that kind of coordinated movement. And if you compare that to a different electrode here in the postcentral gyrus, you'll see that the activity here is quite different. Most of the movement is actually at the level of the lips. The tongue is not doing much, or the jaw, but this is an electrode that is tuned. And we've been able to observe this over hundreds and now thousands of different sensors in this particular part of the brain that are clustered around the central sulcus. And this is essentially a map of different movement trajectories that are important for speech. Jesse Liu and Lingyun Zhao in my lab recently had a study where we looked at the planning of speech sequences, not the control of the movements directly at this elemental level, but really the planning and sequencing of the movements. And they created a task where we had people speak these kind of nonsense or non-word phrases and syllables. The simple ones would be ba ba ba, but then we'd look at complex sequences like ba da ga or ga da ba, and then complex articulations like, for example, blah, dra, glue, really complex in terms of its articulatory movements. And one of the things that Jesse discovered is that we found this dissociation between electrodes along this precentral gyrus that were strongly corresponding to the complexity of the sequence versus the articulatory complexity of the syllable. So one is at the level of sequencing the individual syllables, and the other is about this more instantaneous complexity of the different articulators. And this turned out to be spatially defined. One of the key findings was that there was a cluster of these electrodes that were centered around the middle precentral gyrus, just in front of the area for the dorsal-laryngeal motor cortex, that was very much activated and strongly corresponding with the complexity of the sequences. And if you look at the ventral part of the precentral gyrus, this was corresponding to the articulatory complexity. So we saw this dissociation between sequence complexity and articulatory complexity. The more middle precentral gyrus is very important for sequencing syllables, and the lower is really important for the phonemic or articulatory. And in addition to that, the middle precentral gyrus was strongly correlated with the reaction time when you looked at its pre-speech activity. And then to look at this causally, we stimulated this with transient electrical stimulation. And what we found there was quite interesting. When you stimulate in the middle precentral gyrus, people will have deficits temporarily in being able to speak multisyllabic words like hippopotamus or hospital or catastrophe. This is a spectrogram of someone's speech where stimulation in the middle precentral gyrus kind of creates what we call paraphasias. These are errors in the production, in the sequencing of the speech. And when you're stimulating while people are speaking simple repetitions like pa 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 or t t t, or even simple vowels, there's no interruption of those simple sequences or simple phonetic elements. It really is about the sequencing of this. You can also notice that it's really nothing that we can see in Broca's area that causes these kind of effects. It had been long hypothesized that Broca's area, the human inferior frontal gyrus, was where this sequencing and planning occurs. And we did not see much activity for that or evidence for that at all. This was a case of a gentleman that had a surgery in the middle US that was referred to me by a colleague. And what was really interesting about this was that his surgery was carried out to take out a tumor in the middle precentral area and also the middle frontal gyrus. And it was very peculiar to me when I first heard about it because this was very far away from Broca's or Wernicke's area, the classic language areas that we learn about. But we now know through stroke studies and even our surgical series that this area that we call Broca's area really doesn't often cause speech deficits. And we're starting to learn that the middle precentral gyrus actually is strongly correlated with the ability to have fluent speech. So in this video, you'll see he struggles with the ability to sequence multisyllabic words.
Salpad, Peg Swag Salpad, Max Swag Salpad, Max Swag Salpad,
So this is what we call apraxia of speech. And apraxia of speech is a very interesting and very debilitating condition where someone essentially has a deficit in the coordination and sequencing of words and the planning of speech, but it's not related to the weakness of the orofacial muscles. And they also have totally normal language. They can read and write. In fact, he still works as an accountant, but it's a specific loss of that skilled ability to speak sequences. And so there's this emerging evidence that we have that the middle precentral gyrus is in fact really a critical locus for fluent speech. And this ongoing model that we've been developing, first described by a review in Journal of Neuroscience that we published in 22, but it's really been underappreciated for decades. We've really thought of this area as just quote unquote the motor cortex, but in fact really includes also the premotor functions for the planning and control of speech, the premotor functions that we see in speech. The area that's in blue is what we call the middle precentral gyrus because anatomically that's where it is. You can have direct motor control of the larynx. Stimulating there can induce vocalization. As I described earlier, there's auditory responses in this particular part of the brain. And we now know that it's involved in phonological sequencing. Also, our other groups, our colleagues have described when you're reading, it will actually activate this part of the brain. And then lesions of it can cause a speech syndrome called pure apraxia of speech. So initially what we caught the motor cortex actually has much more complex function and not Broca's area as we once assumed. Some new unpublished work led by a postdoc in my lab named Itzhak Norman is very exciting. He has discovered essentially a fundamental question, which is that we have this distributed orofacial representation and planning area for speech, but how does it get coordinated? Across the lips, jaws, and tongues, there's nearly an infinite degrees of freedom across each of these articulators. How does this become coordinated, both in space and time? And what Itzhak discovered is that there is an intrinsic theta rhythm, a sensory motor rhythm that has been described very well in animal studies. Yuri's work and many others has defined this theta rhythm as a key intrinsic rhythm that governs cognition but also movement. And in this particular work, Itzhak has described essentially how the theta rhythm actually is a conductor for speech movements. So if you look at the coherence of the theta rhythm, it actually increases significantly right before the onset by about 500 milliseconds before someone speaks a sentence. And then once the sentence is initiated, if you look at 25 different electrodes across the speech cortex, the speech motor cortex, it becomes remarkably coherent in this particular frequency band that we can record with a local field potential. These speech responsive sites show an increase in the coherence, suggesting that there is a shared oscillation across this network. And here you can look at the pairwise phase coherence, but then you also look across the group level. There's a striking and clear increase in the theta phase coherence. It's sort of like an internal metronome of speaking rate and timing. And when you look at the different electrodes across the brain during these tasks, they're not everywhere. They're heavily localized to the Rolandic pre- and post-central gyrus, as well as the back part of the superior temporal gyrus. So it's as part of this whole speech network is entrained in a theta oscillation that is regulating the timing of all of the activity, including the high gamma and activity that we see there. So in this particular plot, you're looking at the bandpass theta activity between 6 and 10 hertz of the raw local field potential. The color corresponds to the phase of the cycle of this theta oscillation. What you're looking at here, what we're calling articulatory change, is the peak derivative of the aggregated movement of the vocal tract. And what we had seen in this particular study is that speech is not really a continuous set of movements, but it's actually a pulsatile set of events that are sort of landmarked around periods of when you have a lot of articulatory change and then and not. And this is what these peaks, and these peaks turn out to be completely time-locked and coupled with the ongoing local field potential phase. So basically, the phase of the local field potential actually predicts the timing of these high velocity, high entropy articulatory movements, sort of like a clock that regulates and coordinates our speech movements. So what I've just described is a series of studies that we've done over the last 15 years that help us understand the control of the production of speech. And it's those foundational studies that really allowed us to understand speech as a problem in speech motor control, both at the control of the articulators, the muscles, the trajectories, the phonetic elements, and their planning. And because we were essentially developing a dictionary for all of the different phonetic elements, the consonants and vowels, we decided to begin a series of translational research studies in the lab. These discoveries directly led to the first demonstrations of what's called a speech neuroprosthesis. It's really about this concept of not trying to go after the abstract representation of speech linguistics, but really understanding speech as a motor control problem, similar to the way that the field and brain-computer interface was looking at things like arm and hand representation. Now we were applying to the biology of speech, and it became a critical link at a cognitive and conceptual level to now explore this. One of the reasons that we were very interested in speech is because it allows us to communicate the ideas from my mind to yours and vice versa at a rate that is far faster than any other means, including handwriting, text, typing. Most of the BCI field had been focused on moving cursor controls or handwriting, etc. Extraordinary work, really, and incredible. But speech is in a different category of its own. The way that I'm speaking right now is about 150 words per minute. And this is really, I think, a very important thing. And it's allowed us to transmit information from one mind to another at a high rate, really something important for social cognition, social communication, and civilization. So what we wanted to do was be able to see if this would work in people who are not paralyzed first. We wanted to see if we could use artificial intelligence, machine learning algorithms, in particular recurrent neural network, to translate the activity of speech in people who had epilepsy into words that you can hear directly from the brain. And in this study that was led by Gopala and Josh, they basically built a decoder that translates the brain activity, in this particular case, the high frequency part of the ECOG, into a digital simulation of the vocal tract. As I described earlier, we've been doing a lot of work trying to understand what is the relationship between the representations of movement and the vocal tract. And this part of the brain is not representing text. It's really representing movement and speech sounds. And so we built a decoder that we call bimetic in its approach to really trying to control a digital simulation of the vocal tract through this BCI approach. So we built a speech synthesizer directly to the brain through this intermediate representation of the vocal tract model. And in this video, what you'll first see is the neural activity that is being translated into a virtual simulation of the vocal tract. And then the movements of that vocal tract are then translated into speech that you can hear. And you'll first hear what is decoded by the computer, and then you'll hear what the person actually said into the microphone.
The proof that you are seeking is available in books.
The proof that you are seeking is not available in books.
The proof that you are seeking is available in books. Shipbuilding is a most fascinating process.
Shipbuilding is a most fascinating process.
Shipbuilding is a most fascinating process.
So what you just heard were the two best examples that we had. A lot of this was actually unintelligible. But it was really the first time that we or others have been able to actually see that you can translate this electrical activity, again with modern machine learning techniques, into something that is now borderline intelligible. It was the first time that we could hear words directly synthesized from the electrical activity of the speech cortex. And it was enough for us to go to the FDA and create a clinical trial. And the first participant in this trial was someone who did not have epilepsy, someone who had a severe stroke, a brainstem stroke. His name is Pancho. And about 15 years ago, he had a stroke that essentially left him unable to move his limbs and unable to speak. Absolutely devastating, a near locked-in kind of situation. But he's able to move his neck. In fact, the way he communicates is by using the stick attached to his baseball cap to type out letters and words on a assistive device, which takes him about 5 words per minute. So it's a very kind of painstaking process. But in fact, he's thrived for the last 15 years using this, and it does work. He was the first participant in our study. This is his sagittal MRI. It's a cross-section of his brain. And you can see that in the brainstem is the part of the nervous system that connects the brain to the spinal cord below. And where he had the severe stroke, most of that part of his brainstem is gone. It is essentially reabsorbed, and he basically has very thin fibers now connecting his brain to his spinal cord, and effectively is disconnected from his body. And so what we did in this particular study with the supervision of the FDA is to use one of our epilepsy grids. We attached it to the BlackRock percutaneous pedestal, and they've been an important partner in us in essentially building this hybrid device. And we connected it to a computer that basically translates Pancho's intended speech movements into text that we can see on a screen. This is what the port looks like. Each one of these little gold sensors is connected to a wire that runs underneath his scalp and then is connected to a different part of the speech motor cortex. And so during these research sessions, we can temporarily plug into Pancho's brain and get a direct readout of his intended speech movements, even though his brainstem has been disconnected. And so these are the areas that we found were quite important for decoding speech. This is in the detection algorithm when he's trying to speak. Again, this middle precentral area. And again, this is one of the early important descriptions for us in confirmation. This is a really important area for speech, right underneath the hand knob. And then also classifying different words was really located down in this area right next to the sulcus in the ventral precentral area. These were the first 50 words. This was very modest, to be honest with you, but it was the first time that we or others have been able to achieve any success in decoding sentences. Here we used a limited vocabulary of 50 words. And this is a confusion matrix. If it was perfect, it would be completely dark across the diagonal. But in certain instances, there were really clear confusions, like between the word it and is, and things that were phonetically similar. I've described to you how this part of the brain is encoding phonetic information, and not surprisingly, those word items that are similar phonetically actually had confusion with this model. So this is essentially what we did about 5-6 years ago. He's connected to the port. David from the lab is writing to him. The blinking dots corresponds to the speech detection algorithm, which just detected that he wants to say something, and then classifiers determining what word he's trying to say. With this system, we got to about 15 words per minute with this limited vocabulary. It uses a language model to take the context of the word probabilities and then assemble it into the sentence. Similar to what you have in the autocorrect on your texting. And so we went to the FDA and asked them if we can increase the number of sensors from 128 channels to 253. And we're very fortunate to work with another incredible pioneer named Anne, who had a similar incident about 18 years ago, where she was playing volleyball with her friends, collapsed, and woke up in the hospital unable to move her arms, legs, and was unable to speak anymore. And after her daughter went to college, she moved down to California and participated in our trial. This is a picture of Anne and her husband, Bill. And so what we found was by increasing the electrode number of sensors, we got much better performance. Using this task where we're looking at NATO code words like alpha, beta, Charlie, et cetera, we were able to get to nearly 100% accuracy in decoding these individual words in about a week and a half after starting these experiments. We took the algorithm. After about 10 days, we froze it and continued applied it, actually for several weeks and months, and it continued to have very high performance. And we decided to shoot for some more ambitious goals, doing high performance speech neuroprosthetics across 3 dimensions, from brain to text, brain to control of a voice, synthetic voice, and also brain to control of a digital avatar. So essentially 3 AI machine learning engines decoding brain into 3 different outputs simultaneously. And this is what it looks like when we were able to get about 80 words per minute with Anne essentially doing what we call text copy. This allows us to calculate the accuracy of the speech BCI given a set of prompts. And so in this particular paper, we're up to about 25% word error rate, and the more recent analyses of this data, we can get it down to about 5 to 8% word error rate using new approaches to the machine learning and also signal processing. You can see that she's no longer decoding word by word, but sentence by sentence, and that's much faster. And we built, again, the system that is an avatar. The brain is controlling this digital simulation of an avatar that Anne chose.
I think you are wonderful. Give me a few minutes.
So these were really the first times we tried anything like this. A lot of room for improvement, but again, really just proof of concepts, first demonstrations. And because we're recording from the brain, there's not only processing speech, but orofacial movements. We could decode gestures like puckering the lips, opening the mouth, or even emotional expressions like surprise, happiness, and sad. We've continued to innovate on the technology to try to make it faster. This is an example of what we call low latency streaming brain to voice.
I am sorry, but I have an appointment now.
When the text on the top turns green, it means go.
Let me tell you what I did. Thanks for telling me.
Okay. And Alex, a graduate student in my lab, actually went back to Poncho and built a decoder that can sense if he's trying to speak Spanish or English based on these neural activities from the same brain area. So in this particular video, you'll see him doing Spanish decoding using a Spanish language model. But it's not pre-assigned. It basically is decoding essentially what language he's saying from the phonetic patterns in the given language, and then uses the language model for that particular language. So over the years, it's been a remarkable journey working with a couple of participants, doing brain to text, brain to avatar, facial expressions, in collaboration with my close colleague at UCSF, Karnesh Ganguly, has led really accelerated efforts to also tap into the hand and arm cortex in order to have our participants do robotic arm control. And this has been really an extraordinary time to also be working in parallel with the incredible team at BrainGate, because at the same time that we were making essentially these forays into speech neuroprosthesis, parallel studies through BrainGate using intracortical microelectrodes were being done at Stanford and Davis. And that work is nothing short of incredible. So the goal is to take this out of lab and to make sure that we can get this to clinical practice. Here are some of the things that are important for that. We want it to have high performance. We want it to be easy to use. We want it to be used across multiple indications. and disease severity. I think our group and others have observed that, unfortunately, sometimes with ALS in very, very extreme cases, there's degeneration of these neurons that we want to record from. And we're looking at other strategies, other brain areas in order to do decoding when those motor neurons are degenerated. And we need systems that are fully implantable, fully wireless, that are really easy and safe to implant and explant as medical technologies. The last thing I'm going to finish with is sort of where we're going in terms of the science. We're kind of going back. We've been working on the translation of the BCI, but we're 150% also still committed to the basic biology and really towards a goal within the next 5 to 10 years to understand the cellular base of speech and language. And our group was one of the first to translate the very powerful NeuroPixels technology to humans. We call this project, and we're trying to build a greater consortium around this that we call NeuroPixels for Humanity. But the basic idea is translating this technology so that it can be safe and robust for large-scale human neuronal recordings. These are figures from a recent paper published by Matt and Laura in my lab last year from the auditory cortex, the STG. And so if you look at the response of a single neuron as someone is listening to speech, this is the spike raster for 6 different trials. You can see that it's very well sensitive, very tuned to the N and the junior and the on and the matter, this kind of sound. And in this video, you'll see that this neuron really likes that particular sound.
Junior, what on earth's the matter with you? Junior, what on earth's the matter with you? Junior, what on earth's the matter with you? Junior, what on earth's the matter with you?
So what we've done now is looked at hundreds of different units that are in the STG. A lot of it is confirmed what we knew from the ECOG, but now has added a dimensionality that we've never had, really to understand what's happening at the cellular level, and also the multilaminar looking organization, the connectivity within these microcircuits. These are the rasters of 7 different neurons tuned to these 4 different sentences. You can see that they're activated at very specific times, tuned very precisely. In some cases, very sparsely. In other cases, bursting patterns. Really interesting thing that will take us years to understand. We've also been working with IMEC, the manufacturers of the NeuroPixel probe, to scale this up in terms of how to get more units. Our real limitation in the operating room where we're doing this is time. And so what we've tried to do is increase the number of probes in order to record more units. And this is an example from the ventral precentral gyrus, the speech cortex, production cortex, in some unpublished work led by Quinn and Tommy in my lab. You're looking at the very rich set of action potential waveforms collected by 3 adjacent NeuroPixel probes. And Tommy put together this video that kind of describes how incredible this data is. As Josh was alluding to, for the very first time, looking at human brain cells processing this at a level of scale that we just haven't had before. If you look at the individual units, here are 6 different units corresponding to different words and pseudo words. This is a part of the brain that is processing the phonological aspects of speech production, so it's active during both words and pseudo words. You can see that it's very well tuned across different units. And some of this tuning we can understand a way similar to what we had done historically, that they're tuned to different phonetic features. So if you look at this first neuron, this spike raster, and all the different phonemes, consonants and vowels in English, this is an electrode that is tuned to the voiced consonants. This is one that is tuned to the unvoiced consonants. This is an example of a neuron that is tuned to the sound g and k. This is what we call back tongue. This is an example of an electrode that's tuned to the n and m nasal sounds, something that's inhibited by nasal sounds, and so on. And so this is the first time we've been able to see this kind of selectivity at the single neuron level to the distributional patterns that give rise to human speech. And this is where it begins, but we're at the very, very early experience with this kind of technology. And what gets me very excited about is not what we see at single neurons, but really seeing what we see at scale. So when we look at our data set for the superior temporal gyrus, where we're looking at speech responses to sounds, we're now at about 15,000 cells. You can see it's a whole different data regime, and it's just a testament to how powerful these tools are. So I'm going to conclude here that over the last 15 years, our understanding of the speech cortex has radically transformed from the work in our lab, but many, many other labs, primarily enabled to some degree by the resolution of intracranial recordings. When I started a lot of this work, we had the idea of where these areas were in the brain, but we didn't know sort of the local representations and computations that gave rise to behavior. In the first section, I talked about speech perception, the transformation of speech to words, how we've been able to catalog the selectivity to all of the different consonants and vowels and their phonetic features, as well as the prosody of the voice. But it's not just about the elements of speech. It's also about how that same brain area extracts word level, word form information from the continuous speech stream. And that kind of process is very important to the specialization to our native language, and therefore our ability to comprehend. In the second chapter, I described work that was in the very parallel physiology of the speech motor control, our ability to control the vocal tract to give rise to shaping the breath, the somatotopic organization of different articulators, its dynamics, the discovery of a specialized part of the human brain for control of the larynx, movements of articulators, and a deeper appreciation of the premotor speech planning and preparation functions of the precentral gyrus. These are the premotor functions of it, and how this is becoming temporally coordinated by an intrinsic theta oscillation. We've taken those insights, that basic science and understanding of the speech motor control, to the application of what's called a speech neuroprosthesis, to develop the first demonstration and proof of concept that it's possible, and now been very quickly elaborated by our groups and others, first in 2021, then to high-performance systems across a variety of different modalities, and even doing this in a bilingual context. And then a really exciting next chapter for the lab, digging into the cellular neurobiology of words, using neuropixels, spatial transcriptomics, other methods to understand how computation occurs in the speech and language context. Cortex works on a huge scale, both at single cell and population. One of our goals is to understand how these areas work in a three-dimensional way. So I want to thank the incredible people from my lab, both present and past. These are really the people who have done the vast majority of this work. And it's just a final thought. As I alluded to in the very beginning, none of this work could have been possible without the partnership and voluntary efforts of our patients. Whenever we talk to them about these experiments, I sometimes ask them, why are you being involved with this? Why do you participate? And it's usually universally one answer, which is in order to help others. In all of the research that we do, even in the cases of the speech BCI, we never promise that this will work. It's only that this is part of experimental research to see what will work. This has been probably one of the most inspiring aspects for me in this kind of work, this partnership with our patients, in order to do this work about service and generosity, altruism for the greater good and knowledge. I want to thank my lab for your genius, your perseverance, your creativity. I want to thank my family, Michelle, Dylan, Parker, Stella, for your support and love. I'd like to thank the Gruber Foundation for this incredible recognition. I'd like to accept it on behalf of my lab and also the many patients that have supported this kind of research. Thank you very much.
So we have some time for questions now that'll be moderated by two members of the Gruber jury, John Monsell from University of Chicago and Eve Marder from Brandeis University.
So Eddie, we got many questions during your talk, and a few were asking about American Sign Language. When either hearing or deaf people use sign language, are there representations for words in auditory cortex similar to those seen when hearing people listen to spoken words?
It's an absolutely really interesting question. As many people know, that there are multiple paths into the language system, both in terms of sound, sight, reading, but also through American Sign Language and sign languages in general, the recognition of visual manual gestures. And Matt in our group actually did look at this. We had two particular patients that were deaf signers that we've been able to study very carefully. And what's interesting about that is there's actually a phonology, a sound system that gives rise to the gestures and how people move in particular ways. They're not necessarily in the auditory cortex and in the areas adjacent to that in both the parietal areas and posterior temporal regions.
Eddie, it was a wonderful talk, and I was just curious about whether for people who are hard of hearing, they often find it helps if they can see the person who's speaking to them. Do you have any ideas about how the visual and the auditory get put together in speech comprehension?
That's a great question, and that's about this field that we call sensory motor or auditory visual integration, or multisensory integration. And not work from our group. Some of our colleagues have done incredible work looking at how visual information, for example, you're seeing the movements of my lips, and we actually use that as a kind of Bayesian prior for interpreting the sounds that we hear. And the McGurk effect, where you can have a dissociation of that, is an amazing example of that. This is work by my colleagues Mike Beauchamp at Penn, really one of the world's experts in understanding that multisensory integration in the temporal lobe.
We got a question about whether familiarity matters much for these responses that you see. Do responses differ when playing back a familiar voice versus another speaker?
Yeah, I think it's an interesting question. We haven't necessarily looked at the question of whether the voice quality is familiar. We've always been looking at this from the perspective of whether the language familiar in terms of this native or foreign. But that's a great question for us to look at, is sort of the quality of the voice. And there is work from Taylor Abel and folks at Pittsburgh that are looking at essentially voice identity areas that are related to speech perception of particular voices.
Another question here. What is the relationship between the neural coding of language and other communicative vocalizations, such as laughter, grunting, sighing, etc.?
Oh, great. Well, one thing that we know is that a lot of what is involved in the speech processing is what we call the volitional cortical control. There's a whole parallel system of vocal control that's actually mediated by medial prefrontal areas, like the cingulate, that connect to brainstem structures that are heavily involved with crying, guttural vocalization, other kind of vocal control, but very different than the kind of precise sequencing and control that gives rise to speech. So two different vocal control systems. One is innate, medial, and to some degree a brainstem, and the other is really a cortical one that is highly learned and specialized.
We got a couple of practical questions about the prosthetic, about how long can a neural implant stay in the tissue before the signals degrade, and whether any of those signals could be studied using scalp EEG recordings.
Yeah, fantastic question and very practical one. It turns out that that is probably the most important question that is affecting the translation of this technology, is how long the technology will last. And I think great progress is being made with technology that is placed intracortically in the brain. In order to last longer, we need it to last years and decades, ideally, but also great progress in making safer electrodes that can record from the brain surface at very, very high densities without causing gliosis or injury within the brain. I think both systems ultimately will work, but it's exciting that there's multiple commercial endeavors to figure this out, and time will tell ultimately. The thought of doing this non-invasively, I think, is still a bit of elusive. I think our group and many others have tried this before and may require a lot more data than we currently have in order to make this work. I would never say that this can't happen, but currently it's much easier to be able to decode speech signals directly from the brain itself.
So, given that theta appears to be something like a metronome for speech movements, can speakers voluntarily control the frequency of theta by speaking faster or slower? And also, what happens to theta rhythm during singing or reciting poetry, which might require precise timing?
Yeah, that is a really good question. And so we think that the theta is actually an internal metronome. One thing that we found is that when people speak much faster, they start to skip particular syllables. So even though you're speaking faster, actually we find that the articulatory change is very similar because they're actually still clocked to the theta. So we do think that there's this intrinsic relationship between the theta internal oscillations. And yes, that can shift a little bit, but not dramatically. And when you speak much, much faster, what you'll find is actually people will actually skip articulating different syllables in a way that still allows you to be coupled to this theta rhythm.
We got a question on chunking and the drop in neural activity that happens when words are chunked. How does that work for complex words that might be interpreted as either one or two words? You had an example in your slides, the word judgeships.
Yes. This is a great question and one that we're looking at very closely right now. And for the most part, what we found in terms of this chunking and this drop in the neural signal was really depending on the context of which these words were in the sentence. And certainly when some of these long polyword constructs, composites, complex words were being processed, they were being processed as one unit. We see the drop in the electrical activity at the word ending and not actually in between the subwords within that. So I do think that the context does matter within the sentence. The brain is essentially predicting on the fly using the information that we're hearing and essentially using this as a prior in order to aggregate the information. And what we think is this drop in activity is essentially a resetting of that neural activity and organizing the information in time.
So I just have a question. Do you think that we're going to see cochlear implants as something of a sort of a momentary period in time and would be supplanted by other devices later?
The cochlear implant is an amazing example of one of the first essentially brain-machine interfaces. And that was many decades ago now, and a real miracle of science and technology. I think in the future, it works quite well already, but there may be options in the future to enhance hearing directly from the cortex itself. There are many people who actually have injuries to the auditory nerve who are not candidates for the cochlear implant, so there's ways that we may be able to brain-computer interface better to the brainstem or some of the subcortical nuclei or even directly to the superior temporal gyrus. So that's something we're very interested in pursuing.
We have one last question to end the session, and that's about the amount of variability between different subjects and whether you've had a chance to actually explore that or whether you've seen obvious signs of real differences between individuals.
It's a great question, and we do see huge variability across individuals. I think the localization of the function that I've described today is largely the same. Everything that we see in terms of the activations are in the same ballpark. But if you look at individuals, there is quite a degree of variability in terms of the localization of the specific tuning and encoding. Everyone's brain so far that we've looked at, actually none are identical in terms of their pattern. So the broad ballparks are very similar, but at the individual level continue to be highly specific. Thanks, John. Thank you. Thank you so much.
Well, I want to thank Dr. Chang for a truly extraordinary presentation and congratulate the Gruber Foundation on an inspired choice. This concludes the Gruber program. Thanks again, Eddie.